Okay, so now we're going to start with chapter 14. Chapter 14 is all about the, am I sharing the correct screen? I'm sorry. Hold on. Chapter 14 is all about basically mutations, DNA repair, transposons, which are really fun. Basically is your DNA jumping from one part to it, of your DNA to another. It's insane. And so it's a really important chapter. So mutations are critical, right? We study them not just because with mutations, this can bring about different kinds of disorders and diseases, genetic diseases, but this is also the source of allelic variation, right? This is how adaptation and just basically how we evolve and change and respond to our environment, how it happens. Our program has to be changed. Our genetic program, our genetic encoding has to be changed in order to adapt in some way, right? So it's a source of new alleles. However, we can obviously have negative consequences. So this can cause things like cancer, cell death, genetic disorders. So very negative things depending on the mutation and where exactly it happened, right? Because as we're going to see, there's going to be mutations that happen in purely like benign, kind of useless. I say useless in quotations because it's just honestly areas of the DNA that we still don't really know or understand very well. But even in mutations happening in those areas don't seem to cause that many problems, thankfully. So there's mutations happening in this kind of non-coding useless DNA, and other mutations happening in proper like coding regions, or even mutations happening in regulatory sequences. We saw a little bit about, we talked a little bit about regulatory sequences in chapter 12, I believe. When we're talking about transcription, we saw what the gene looks like and how the gene has upstream of the actual coding sequence of the gene, we have the promoter. That promoter is part of the regulation of the gene's transcription, and therefore it regulates whether the gene gets transcribed and made into protein or not, right? And so we have other sequences as well, like enhancers, silencers, and a variety of other kinds of regulatory sequences in a gene. And so if the mutation happens there, the results can be very bad, right? It can mean that the gene gets expressed all the time, or that the gene is never expressed. It's always inactive. It's always inactive. So mutations are huge to study. And of course, they're also key players in how we actually, in how we analyze organisms and learn about genetics. We saw in countless different experiments, but in order to figure something out about a protein or about how this particular DNA works, we need to mutate it a lot, right? So mutations are key players in genetic analysis techniques. Think of anything like the earliest one we went over was the Neurospora, right? We mutated, not we, but like Tatum and beetle. They basically bombarded Neurospora with UV rays to make new mutants, right? So what is a mutation? A mutation is an alteration in DNA sequences. This includes things like a modification of the actual base, so a substitution, right? An insertion, a deletion. This could be a point mutation, so that's just like when one nucleotide is changed, right, or inserted or deleted. And then it could be even a major chromosomal one where the entire bigger segment of DNA that makes a chunk of the chromosome is actually altered. They can occur wherever on the DNA. The extent of the mutation changes on the organism, the extent of the mutations change on the organism's characteristics depends on which cell type has a mutation and the degree of the mutation. Classification is based on several different schemes, but as we'll see, so we're going to look into the different degrees of mutation and where the mutations can happen, and then we're going to look at these different kinds of classifications. And when you see the classifications, you'll notice that they're not all mutually exclusive. There's a lot of overlap, right? Let's start looking at that. So classifications based on molecular change. So there's classifications based on molecular change, classifications based on phenotypic effects, on where the actual mutation occurs, right? So lots of different kinds of classifications. The first one is based on the type of molecular change. So basically, it's about how big this mutation is. And when I say big, I'm not meaning like how much of an effect it has, just like where and how many bases it gets involved. So point mutations are just basically when one base pair out of the entire DNA sequence, only one of them gets substituted, okay? So these are basically alterations. But there's a lot of point mutations that happen with DNA replication, with even things like editing, right? So the point mutations are when we have that one little mistake. Frame shift mutations are things that are mutations that shift the reading frame. So we know that the reading frame is established right at the AUG at the start codon, okay, and then from there we read its 3 codons at, not 3 codons, but 3 nucleotides at a time. So codon to codon to codon. Now, if we have an insertion or a deletion of anything that is not a dividend of 3, essentially, we have a frame shift mutation. Okay, so this results from the insertion or deletions of a base pair. And it occurs when any number of bases are added or deleted, when except for multiples or dividends of 3, right, which would reestablish the initial frame of reading. And this is because codons are triplets. So you change the codon frame, and then the reading frame, and of course you would change the entire sequence. It's a humongous, it's really, usually these frame shift mutations are quite disastrous, okay? So like I said before, point mutations, there's a few different kinds. They change one base pair, right? So the point mutations can be a missense mutation. This is when that one base pair results in a completely new codon, obviously. And then this new codon actually doesn't code for its original amino acid. Okay, so it codes for a completely altered, changed amino acid within the protein codon portion of a gene. So it would be like changing, I wish I had the genetic code in front of me. Do I have it somewhere down here? I do not, sorry. I do not. That's kind of annoying. Because if I open another PowerPoint, you can't see it. So the basically the missense mutation, a codon is changed, right, and is changed to another type of codon that encodes another amino acid. I'm going to see here. I'm going to stop the share real quick. Here we go. So say I have my my codon ACU, right? ACU normally codes for threonine, but say this initial A gets there's a point mutation. This initial A is now changed into another C. So I now have a CCU, right, codon, and that's going to give me a proline, right, which may or may not have completely, in this case, does completely different characteristics, and therefore change the polypeptide chain. Okay, so that's called a missense mutation. We also have a nonsense mutation. Nonsense point mutations are when your codon is changed in such a way that it becomes a stop codon. So, for example, UAU normally codes for tyrosine. Let's say, for example, my U here there's a point mutation and it gets changed into another A. So I now have a UAA. And so now this codon actually encodes for a stop signal. And so this could really truncate your protein prematurely and give us a truncated protein that does not work, right? Depending on where this nonsense mutation actually came about. In addition, you also have silent mutations. Silent mutations, I believe I'm still on the correct. Yeah, silent mutations. Silent mutations are when the codon changes. Again, you have that point mutation. However, it's still the new codon that arises still codes for the same amino acid. And this is possible because of the genetic code being so degenerate, right? So if we see here, we have leucine, CUU can code for leucine. If I change this last uracil to a C, to a cytosine, I'm gonna have CUC. No problem still there, and that's I still get leucine from it. Okay, so we have these 3 types of point mutations, so it's it just basically depends on what the codon becomes in the end, right? Base substitutions, it depends, like basically base substitutions, you can either have transitions or transversions. Transitions are when we go from pyrimidine to pyrimidine. So we have a thymidine or a cytosine becoming a cytosine or a thymidine respectively. Okay, so a thymidine gets point mutated into a cytosine, or a cytosine gets point mutated into a thymine. Okay, and then that's known as a transition, as is going from a purine to a purine. So a guanine gets point mutated to an adenine, right? Or an adenine gets point mutated into a guanine. Remember, purines, like this is all remember what pyrimidines and purines are. Purines have 2 ring structures, pyrimidines have 1. Okay, when we go from purine to pyrimidine, this is known as a transversion. And so we would go from A to T or G to C, right? This is known as a complete transversion that has occurred. So that's basically the different kinds of substitutions that we can have. And then we'll look at frame shift mutations, which are caused by insertions or deletions, specifically bases of like either 1 or 2 bases, right? If we have an insertion of 3 bases, that doesn't cause that big of a deal because you're basically adding a new codon, but you're still keeping and maintaining that reading frame. But when we insert just 1 or 2, or when we delete just 1 or 2, we're going to have a frame shift mutation because the entire reading frame shifts. So the codon reading frame shifts except in multiples of 3. And you can basically see the effects of the reading frame. Oh, goodness me, sorry. The effects of the reading frame from this particular picture, this particular image. So a substitution would be something like, instead of saying the cat saw the dog, it'd be the bat saw the dog, the cat saw the hog, the cat sat the dog, right? So still significant, we're changing the entire meaning of the sentence. If we lose 1 letter, so there's a deletion of a base or a letter in this case, it would now completely make no sense. So we get rid of the C, and now that remains the same, but now it's like at, out, head. I'm not going to even try this, but can you see how by removing the C, all the other codons, all the other 3-letter words, but like after the actual removal of the base become affected? Same thing with the insertion, all the codons after it become affected and it becomes a jumble of things, right? And there's usually a very high chance that you get a nonsense mutation with this. You get a mutation that encodes a stop codon. So in addition to having a frame shift, you're also most probably going to gain a truncated protein. Okay. Another thing that we classify our mutations on is the phenotypic effect. So we could have loss of function, gain of function, morphological, nutritional, behavior, regulatory. So basically what they do to the phenotype. Loss of function is when our mutation heavily reduces or sometimes even completely eliminates the function of a recessive of a gene product. This is most of the time a recessive mutation. Okay, so with recessive mutations, you need 2 recessive mutations on the same gene in order to bring about that recessive mutated phenotypic effect. It can also be a dominant mutation. Here, the result of the mutant phenotype is present even if the wild type is present, because the dominant is going to be it's always present in the phenotype. So the reason why this comes about, right, the reason why we have these dominant mutations is because of 2 different effects. It's because of these 2 different effects. Once we have, one of them is haplosufficiency. So this is when that single wild type copy that you had, when you put it against, when you compare it against the dominant one, right? We have, in this case, you'd have 1 wild type copy and then 1 dominant mutant copy, 1 other dominant mutant allele. That single wild type copy does not produce enough of the gene product that is necessary or to bring about the wild type phenotype. Okay, so you would normally need 2 wild type phenotypes, 2 wild type alleles to bring about that wild type phenotype. And then because of that dominant mutation, it doesn't work out just because it's insufficient. So that's known as haplosufficiency. We also have dominant gain of function mutation. This is when the gene product basically through the dominant mutation actually gains a new function, right? This function could be good or bad, doesn't matter. If it's bad, it's called the dominant negative mutation, positive, dominant, positive mutation. So the dominant negative mutation directly interferes with the function of the product of the wild type allele. So, and obviously it will give you a bad mutation, pretty much a bad mutation. So here you essentially, with these recessive and dominant mutations, if they're loss of function, you reduce or eliminate the function of the gene product, right? And it's hard to think about. Obviously, it's easy to consider this with recessive mutations because you just don't have the normal allele, the wild type allele to make the gene product. But it's harder with dominant mutations because with the dominant mutations, the wild type is present. But just know that either through haploinsufficiency, the wild type just doesn't make that allele doesn't encode or enough, doesn't produce enough gene product, or the dominant mutation has now gained the function of basically shutting up the wild type's protein and interfering. Okay, so we also have non-mutations, very similar to loss of function, but here there is a complete loss of function. Okay, so there's no amount of like small reduction or something like that. It's a complete loss of function. Visible mutations are mutations that are obviously visible. So I'm going to be like basically reading these things out. They're pretty self-explanatory. They affect a visible phenotype. So this is anything that you can actually visibly see. So, you know, I don't know, like the peas in Mendel, right? You could see the different colors and all that stuff. It's most easily observed because they affect the morphological trait. You also have nutritional mutation and biochemical mutations. Obviously, these mutations can have an effect on the morphological appearance, but they stem from nutrition, like a nutritional loss. So by saying nutritional loss is that if the organism can no longer synthesize a particular nutrition, like a vitamin or an amino acid or a nucleotide even. Okay, so that would be the nutritional mutation. That's a lot of those were seen, like remember when we had bacteria growing in incomplete media, or even that neurospora fungus growing in incomplete media, that would be characterized as a nutritional mutation. Biochemical mutations, you're just mutating something, a gene that encodes an important enzyme in a biochemical reaction. So it affects the function of the protein and can impinge well-being of the individual. Behavioral mutations, they affect behavioral patterns, difficult to analyze, because a lot of times they don't really manifest themselves like, you know, physically. It's more like internally behavior. So a lot of these are kind of, again, you won't see, there's, you know, they're kind of blurry. The lines between these mutations are kind of blurry, and sometimes the different classifications and categories just merge and kind of overlap each other. So then we have mutations that can occur in the regulatory regions of the genes, right? So some regions that are important for transcription initiation, like that promoter sequence, regions that are important in mRNA splicing. So even introns, so in eukaryotes, introns are technically intervening sequences that get spliced out, right? Mutations in the introns can still have a really big effect. Maybe a mutation changes a particular, like the branch point or the adenine or something on the, you know, the 3' adenine or whatever on the intron, and therefore the intron no longer can be spliced out. That's going to be bad. And then of course there's also now studies showing that introns are really important in regulating how much the gene is expressed. So how much the gene is expressed, and then what the gene actually becomes in the end, like in terms of mRNA. And also things like mRNA stability. So there's a lot of regulatory regions on your DNA that regulate a specific gene. And if mutation happens within this regulatory region, we can have an inappropriate regulation and control of our gene. So inappropriately active, right? So too active, or inappropriately inactive. So when we need the gene to be expressed, it'll not be expressed. Yeah. We can also have lethal mutations. These ones we've actually already kind of talked about in the past. Lethal mutations interrupt an essential process and result in death. So a lot of these will basically lead to a specific genetic disorder, like a genetic disorder. So something like Huntington, right? So that mutation that causes in the Huntington gene, in the HUT1 gene, is basically considered a lethal mutation because it does result in death. So lethal mutations, don't think that you die on the spot. It doesn't necessarily mean that. It also is dependent on when the gene is expressed. So kind of back to that regulatory importance, right? When the gene is expressed. So that's why Huntington, it's lethal more towards kind of older age. We also have conditional mutations. So these ones are the mutations that are dependent on the environment. So they're not visible, they're not active, right? Unless a specific permissive environment is put on them. Under restrictive environments, they don't even show up. So they can't even be detected under certain conditions, under the normal conditions. So like this would be, remember the Himalayan rabbit in the lecture, we went over that. And so how just depending on the different temperature on its skin, it has either black fur or white fur. Okay, so a good example, and so that's a good example actually of a temperature sensitive mutation. So the mutation is either like permissive under specific temperatures and then restricted under other temperatures. We also have neutral mutations. So most of our mutations actually are neutral. And this is when they happen in a part of your genome that is not necessarily important. So it's not part of the regulatory sequence of your genes, nor is it part of the actual coding sequence of your genes. So it kind of basically falls in that excess junk DNA drawer, that seemingly doesn't do anything. So this is basically neutral mutations, and when they happen there, they don't really change us. They have no effect on the gene products. Classification is also based on the location of the mutation. So location, if it's autosomal, it happens on the autosome chromosomes, right? X or Y-linked, X-linked mutations, they happen on the X chromosome. Y-linked mutations, they happen on the Y chromosome. Somatic mutation, this would be the place and time where I would basically ask you guys what these are because it's in the name, but I'm just gonna say you. So somatic mutations, right? These are just mutations that happen in your somatic cells, which are basically all of your cells except for your gametes, right? So all the cells that, except for your gametes. So when a mutation happens here, right, you don't pass it on to your offspring. It's not heritable. So it needs to occur in the germline cells, right, in the germline mutations or in the germ cells in order to be able to be passed, to be present in your gametes, in your sex cells, so then pass them on to your offspring. So germline mutations happen in germline cells, and they occur in gametes and are inherited. This potential to be expressed in all offspring cells. Somatic mutations are vastly different. In fact, actually, a lot of times when you have a somatic mutation, it's usually very hard to notice because it would be just one cell that has this mutation out of all the cells in your body. In order for the somatic mutation to actually have a really drastic effect on you, it would have to happen kind of in pre-development time. So right when you're going through development where you have undifferentiated tissue, right? And at that point you get a somatic mutation, all the cells that come from that undifferentiated tissue will then have and hold the mutation. So so at that time, it has biggest impact if it happens in early development. That's what I was talking about. So when a recessive autosomal mutation occurs in a somatic cell of a diploid organism, it's unlikely to result in a detectable phenotype, especially since it's diploid, right? You have the other alternative allele. The inherited dominant autosomal mutations will be expressed phenotypically in the first generation. X-linked recessive mutations arising in the gametes of a homogametic female may be expressed in a hemizygous male offspring, right? So it just basically depends on where it occurs. And then we also have spontaneous or induced mutations. These can every single classification we've done so far, they can overlap to some degree. So spontaneous mutations happen naturally and randomly. This is usually through normal biological processes like normal DNA replication, right? Normal DNA editing, like RNA editing and post-transcriptional modifications. So just normal processes. This is usually very accidental. So there's no specific agent that is specifically, egging this on and making more spontaneous mutations happen. In fact, a lot of these spontaneous mutations we try to avoid, right? Thanks to that proofreading mechanisms that we have in our DNA polymerases, we have very little amount of mutations that actually do arise, right? So sometimes, apart from considering the fact that the DNA polymerase itself has very high fidelity in inserting the correct base, when it does mess up, it usually proofreads. And so basically the error rate, the mutation rate, which is the likelihood that a gene will undergo a mutation in a single generation or in forming a single gamete, okay, that mutation rate significantly decreases. I believe it's like 1 every million base pairs in a human. That's how many, every million or every billion, don't remember, but very small amount of errors actually occur. We also have induced mutations, and this is what we've used to basically study a lot of organisms in the laboratory, right? When we specifically mutate them. So we bombard them with X-rays, with UV radiation, irradiation, all these things to basically cause them to gain mutations. So this is when you have an influence of an extraneous factor. right? So mutation rates, the rates of spontaneous mutations vary among the loci in different organisms, right? So the mutation rates actually can even vary in different areas, like in different parts of the genome. There's genomic parts that are known as mutation hotspots that actually mutate more rapidly than others. And then there's also between different organisms, right? Like, yeah, between different organisms, there's a difference in mutation rates between a species, like within a species, right? There's all sorts of things. It's unclear when such variation occurs in mutation rates. Usually, spontaneous mutation rate is low in all organisms. It may in part be related to their DNA repair and proofreading system efficiency. Okay, so we've kind of already talked about proofreading systems and how they help. And then later on in this chapter, we'll talk about DNA repair. Variations between genes in a given organism may be due to inherent differences in mutability in different regions of the genome. There's certain regions, like I said, are called mutation hotspots, and here they are highly susceptible to mutation. Okay, so this is just kind of tells you a little bit of the rates of the different spontaneous mutations in different organisms at different loci. So that's basically where I'm going to stop for now, and we're going to continue on in the next. I'll see you guys in the next video.